Hello. Thank you for watching today's presentation on improving translational relevance with humanized NSG mice. My name is Elizabeth Axton, and I'm delighted to share this educational information with you to support your efforts to achieve research excellence. If you have any questions about the content of this presentation, please contact us using the information on the final slide of this presentation. Before getting started with today's presentation, I wanted to share with you some information about the Jackson Laboratory. The Jackson Laboratory is a nonprofit biomedical research institution. We have our own research staff with their own private funding means, such as NIH grants, that investigate the genetics and biology of human disease, partly using the laboratory mouse. We also provide scientific services and resources to investigators around the world. In addition to our clinical and research services that some of you may already be familiar with, we also have some freely available resources that I'll touch on in more detail shortly. And finally, here at the Jackson Laboratory, we are very committed to education outreach. In addition to webinars such as this one, we also sponsor traditional conferences, hands-on workshops, and educate students of all ages through courses, internships, and other programs varying from high school internships to postdoctoral assistantships. The Jackson Laboratory is the world's largest repository for mouse models of disease. We are NIH funded to import and cryopreserve as many mouse strains as possible. Among the more than 11,000 strains that we distribute at both our Bar Harbor and Sacramento facilities, over 1,500 of our strains are maintained as live colonies. Jack's mice are the best characterized and referenced mice in published research, and much of this can be attributed to our rigorous animal health standards and unsurpassed genetic quality. What many investigators may not realize is that the use of our well-characterized and heavily referenced common inbred strains, for example, C57 Black 6J or Bulb CJ, support the development, collection, and distribution of the specialty mutant mouse models, as well as provides funding for our freely available resources. Some of our freely available resources include our Jack's Mice database. For those of you who have looked for available mice on our website before, you may have ended up on our Jack's Mice database, which has data sheets for every strain in our repository. While your initial goal may have been to see if the colony we have is live or find the price of the mice, if you spend a bit more time on the data sheet, you will see that we have a ton of other useful information. For example, genotyping protocols that we use, phenotype information associated with the strain, development, genetics, and even a list of helpful references that utilize that particular strain. Next, we have the Mouse Genome Informatics database. And this is a great resource that includes information not only about mice from Jack's, but also mice from other repositories and other investigators. I like to think of this database as a one-stop shop for all things mouse genetics. It's a really useful way to search for strains that carry a particular allele, and if they are available through any public repository, Jack's or otherwise, you can link out to those resources on the allele detail pages. The Mouse Phenome database is a collaborative collection of baseline phenotype data for some of our more popularly used strains. Some of the other resources that we have include the Jack's Clinical Knowledge Base and the Mouse Tumor Biology database. Now again, all of these are free online resources available to you to help with your research needs. In today's presentation, we're going to start with an overview of the immunodeficiency of the NSG strain, which makes it a unique host for humanization. We will then cover methods of humanization of NSG and its variant NSG-SGM3, including humanization with CD34 hematopoietic stem cells and peripheral blood mononuclear cells, PBMCs. We will then investigate human immune function in humanized mice and highlight some recent research examples using these mice. And finally, we will end with some resources for you to utilize in your research. Let's start with an overview of NSG mice. NSG stands for non-SCID gamma, and these three components are what make this strain immunodeficient. NSG is based on the non-obese diabetic genetic background. Now, this is an inbred, just like C57 Black 6J, bulb CJs, and others. NOD does not have C5 hemolytic complement. They also have reduced dendritic cell function and defective macrophages. Now, these are not completely absent. However, they do have reduced response to challenges. Now, finally, they have a unique SERP alpha allele, which increases the affinity for human CD47. This makes the NOD genetic background optimal for human hematopoietic stem cell engraftment. Now, interestingly, NOD stands for non-obese diabetic, and these mice do develop autoimmune type 1 diabetes. However, when we introduce a SCID mutation, it prevents the development of diabetes. In addition, the SCID mutation impairs development of mature B and T cells. Now, finally, IL-2RG deficiency eliminates signaling from 6 distinct interleukins listed here, and it also blocks natural killer cell development. So together, these three components accelerate the ability of NSG to engraft human cells. Now, one consideration is that the SCID mutation results in radiation sensitivity. So if you intend on using radiation as a therapy, you may want to consider using the NRG, NOD-RAG gamma, as an alternative to NSG. Now, I find it helpful to view these immune components visually. And so to do that, let's start with a brief overview of the immune system. The immune system in mice and humans is composed of the innate and adaptive components that work together to mount a defense. Now, the innate immune system could be considered as the front line of immunity, an immediate barrier to pathogens or viruses. This is composed of the complement system, which lyses pathogenic bacteria, dendritic cells, which are the antigen-presenting cells, macrophages, which can engulf dead and dying cells and carry off bacteria and pathogens, granulocytes, which are part of an allergic and inflammatory response, and finally, natural killer cells, which are a very important part of this system. These are the cytotoxic cells that can attack and kill invading pathogens. Now, the adaptive immune system is a more specialized system that can target and eliminate pathogens. Now, B cells and T cells work closely together to mount defenses via several mechanisms, with one of their major functions being to produce antibodies. To tie this all together, cytokines, primarily interleukins, are secreted proteins that facilitate the crosstalk between both branches of the immune system. In order to have successful engraftment of human cells in a mouse, you essentially have to wipe out the entire immune system. In NSG mice, you can see that there are defects across the board, leading to severe immunodeficiency. There are virtually no functional complement system or natural killer cells, and really the only remaining innate immune system is portions of macrophages and granulocytes. On the adaptive side, there are no B cells or T cells, and therefore no antibody production. Now, these components together make the NSG mice a great host for humanization. Next, let's talk about options for humanizing mice. The first option is humanizing NSG mice with human CD34 hematopoietic stem cells, which I'll refer to as HSCs. This option is suitable for long-term studies in the context of a human immune system because the mice do not develop graft-versus-host disease. The next option is humanizing mice with functionally mature peripheral blood mononuclear cells, or PBMCs. This is a good option for studying graft-versus-host disease because mice develop graft-versus-host disease within several weeks. A third way is called a BLT mouse. BLT stands for blood, liver, and thymus. You can take HSCs from the fetal liver and thymic tissue and engraft them in the host mouse, as well as with HLA-matched HSCs. This option is particularly useful for some applications, including HIV research. However, the strain does develop graft-versus-host disease. Now, the mouse models with the asterisks are the ones that are currently offered at the Jackson laboratory. We are not currently distributing the BLT model. For the purposes of this presentation, I will be focusing exclusively on the mice humanized with CD34 HSC because of their broad scope of research applications. Now, of note, we now have another webinar solely on the use of humanized PBMC mice for the study of graft-versus-host disease. For generating CD34 humanized mice, we can do intracardiac injection in newborns. However, this is technically challenging and there are lower levels of survival. So we prefer to wait until 3 weeks of age and do intravenous injection to promote survival and promote animal welfare. Our standard offering is in females, given that they engraft more efficiently than in males. While human HSCs can be sourced from different tissues, at JAX we use exclusively human cord blood. Preconditioning treatment in the form of irradiation is necessary for engraftment. Now, this is necessary to reduce the host response to the donor human cells. The engraftment of human cells does take time. We do irradiation and tail vein injection at 3 weeks of age. Then the cells need to first differentiate, fill out into the periphery, and develop functional human B cells and then T cells. So this takes approximately 3 to 4 months to have full engraftment of human cells. Now, one benefit of purchasing these humanized mice from JAX is that we are constantly doing engraftment and aging up these mice. So instead of waiting 3 to 4 months, you can typically receive mice in just a few weeks. I'd next like to give you a brief overview of hematopoiesis because it is essential to understanding the characterization and use of humanized mice. When we engraft with CD34 HSC cells, we have differentiation into multi-potential stem cells, and from there we have multi-lineage differentiation. We can generate lymphoid progenitor cells, which leads to natural killer cells, B lymphocytes, and a couple different types of T lymphocytes. You also have the development of the myeloid compartment, including monocytes and macrophages, among others. Now, as you can see, mature cells have different markers, such as CD56 for natural killer cells, CD3, CD4, and CD8 for different types of T cells, CD19 for B cells, and CD14 and CD33 for monocytes. The nice thing about this is that you can measure these markers with flow cytometry to understand what types of human cells are developing in humanized mice. In this next section of the presentation, we will investigate the human cell populations that develop in CD34 humanized mice. To summarize our findings to date, we have found that the NSG mouse enables efficient engraftment of human HSCs and permits differentiations of major cell types. This includes those of the myeloid lineage, including erythrocytes, megakaryocytes, monocytes, dendritic cells, and granulocytes, as well as those of the lymphoid lineage, including T cells, B cells, and natural killer cells. In addition, there are some mucosal and adaptive immune functions. So let's dig a little bit deeper into that data. Let's first start with some data from the literature regarding humanization of NSG mice. Now, staining for human CD45 allows for us to quantify the percent of human donor contribution to the total pool of CD45 cells. Now, these three figures are in first the bone marrow, spleen, and peripheral blood. We can see that in each cell type, there is engraftment of the human cells, as indicated by human CD45. And this ranges from about 40% at the lower end all the way up to almost 100%. And this is pretty consistent across the 3 different cell types. Now, within that CD45 population, we can look at what proportion are T cells, which are CD3, B cells, which are CD19, natural killer cells, which are CD56, and also the myeloid cells, which are CD33. Now, as you would expect, in the bone marrow, you have a higher proportion of CD33 myeloid cells compared to that which is seen in the spleen and in the peripheral blood. You may also have noticed that there are higher T cells and B cells in the spleen and peripheral blood in comparison to the bone marrow, which is what you would expect to see. Now, one additional thing that I want to highlight here is that in all 3 of these compartments, we see low engraftment of these CD56 natural killer cells. That's across all 3 cell types. And we are going to be discussing this in a bit more detail later. So together, these values indicate that you have engraftment of CD34 cells and subsequent multilineage differentiation. The next question that you would need to ask yourself is, what about the T cells? Are they mature or immature? Now, they should initially develop as CD4 and CD8 double positive, but then differentiate into CD4 or CD8 single positive and migrate into the mouse thymus and into the peripheral blood and the spleen. We can see here in the thymus that there is a population that is singly positive for CD8, another population that is singly positive for CD4, and there's still a pool of doubly positive cells that will likely continue to differentiate. Now, one important factor in humanized mice is the human donor itself. Donors have significant variability in their engraftment rates that ultimately impact their relative immune cell populations. Here we took 3 different human donors and engrafted NSG mice and analyzed them 12 weeks later. We see that all 3 of these donors have CD45 values that are relatively similar. However, we can see striking differences in differentiated immune cell populations. In this case, most remarkably in this population of B cells, and also in this population of monocytes. Now, this example is specifically in the bone marrow, but we can also see the same thing in the spleen. Here we have more lymphoid engraftment, as evidenced by the relatively higher proportions of B cells and T cells, but we still see across the board significant donor-to-donor variations. And finally, here are the same 3 donors as seen in the peripheral blood. Now, the reason why I'm highlighting that there are donor-to-donor differences is that this needs to be accounted for in your study design when using humanized mice. One way that we recommend to account for donor variation is to select multiple human donors for your experiments and spread them across your multiple treatment arms. This will help improve the reproducibility and robustness of your experiments with humanized mice. Another experimental consideration is that the myeloid population's values that I showed you in the last few slides are across the board lower than what you would expect to see in humans. This is because you need human-specific growth factors in order to facilitate myeloid cell development. While the NSG remains the gold standard in reconstitution of the human immune system, primarily because of the functional human T cell responses, it is limited in myeloid lineage development, as we just saw in the last slide. Now, this led to the development of the NSG SGM3 strain. Dr. Connie Eves developed 3 co-expressed transgenes to this effect, and it was bred to the NSG strain. The strain was shown to have improved acute myeloid leukemia, or AML, engraftment efficiency. Then in this Billerbeck publication, it was demonstrated that it improved human myeloid cell development after human stem cell transplantation. So we imported the NSG SGM3 strain into JAX and tested it into our own facility. We took female mice from the NSG and NSG SGM3 strains and irradiated them. We engrafted them with approximately 130,000 cells per mouse, all from the same human donor. We then collected peripheral blood over time and did flow cytometry analysis for several immune cell populations. Now, in this first graph, what you can see is that the NSG SGM3 here in red have higher and earlier engraftment of human cells as indicated by human CD45. However, you can see over time, and about around 24 weeks of age and onwards, the total engraftment is similar between the two strains. Where it really gets interesting is when we look at the individual immune cell populations. In this example, 18 weeks post-engraftment. Now, throughout this slide, the NSG is in green, and the NSG SGM3 is in pink. Now, in addition to having greater total human cell numbers here, we can also see that there are higher myeloid cells in the NSG SGM3 mice. B cells are somewhat equivalent, perhaps with faster development in the NSG SGM3, but over time they equalize. We can also see that there are higher T cells in the NSG SGM3 compared to the NSG. If we look a bit closer at the T cell population, we can see that there is greater expansion into the CD4 helper cells, as well as into the CD8 cytotoxic T cells. So if you're still trying to decide if T cell mediated response is the primary driver for you, then likely the NSG would be suitable for your research. If regulatory T cells, myeloid cells, or dendritic cells are the key, then I would suggest looking into the NSG SGM3 mouse instead. So I've just shown you that humanized mice develop populations of human immune cells. What we haven't covered yet is whether these cells are actually functional. We are going to cover both innate and adaptive immunity. And once again, here I'm going to focus on CD34 engraftment in NSG because that is the most commonly used platform and where the vast majority of published data is on. Let's start with the myeloid cells. In this example, researchers took NSG mice engrafted with CD34 and first confirmed successful engraftment. In this first panel, these researchers isolated myeloid cells from either the lung or the bone marrow and put them in liquid culture and exposed them to fluorescent beads. When you increase the temperature up to 37 degrees, you can see that the myeloid cells phagocytose those beads, which is evidence of a functional myeloid compartment. In addition, we take engrafted mice and inject them with lipopolysaccharide, LPS, which is a molecule found on the cell wall of bacteria that binds to toll-like receptors on myeloid cells. We see that the LPS induces cytokine release, indicating a human pro-inflammatory response. Now, we can see this across multiple cytokines with human TNF, interleukins 6, 8, and 12, among others. Now, in some additional data that isn't shown here, you can also take the isolated myeloid cells, put them in liquid culture, and expose them to interferon gamma and salmonella, and you can get the myeloid cells to phagocytose and kill the salmonella. So when you put it all together, these results indicate that these are functional myeloid cells in the humanized mice. Now, those figures were all from published literature. We were able to recapitulate these findings in our own facility in Sacramento with our own in vivo services group. When given an LPS challenge, there was a human pro-inflammatory response against LPS, as indicated by increased abundance of TNF-alpha, interleukin 6, MCP1, and IL-1B. Another component of the innate immune system are the natural killer cells. Interestingly, while natural killer cells are developed in humanized NSG mice, they require human-specific growth factors, notably human IL-12 and IL-15, to become functionally mature. Now, while you can find functionally mature cells that are NKp46 positive and CD56 positive, most of the cells in the spleen and lung are NKp46 positive, but CD56 negative. This indicates that they are not functionally mature. However, you can isolate the natural killer cells and culture them with growth factors, such as IL-12 and IL-15 or polycitidylic acid for full development. If you introduce these natural killer cells to target cells with non-self-displayed molecules, for example, these K562 or CEM cells, you will see an immune response. For example, here we have untreated natural killer cells, as indicated by the red arrow, and when we culture them with IL-12 and IL-15, as indicated in the blue arrow, we see an increased response to those target cells, as evidenced by release of both interferon gamma and the degranulation markers CD107. Now, you may be interested to hear that we have a brand new model, which is the NSG IL-15. This has increased development of natural killer cells after being engrafted with CD34 cells, and this is now available as a host for humanization from JAKs. Let's move on to the adaptive immune system. Humanized CD34 mice do have functional T cells. If you isolate CD4 and CD8 positive T cell clones derived from the spleen of humanized mice, you can culture them with allogeneic non-self or syngeneic self target cells to assess their activity. For human CD4 T cells, we would expect to see that there would be cytotoxicity of those allogeneic target cells with activity blocked by class 2 monoclonal antibodies. With human CD8, we would also expect that there would be cytotoxicity of allogeneic targets, however, with activity blocked by class 1 monoclonal antibodies. We analyzed the cytotoxicity response with chromium release assays to validate this in humanized NSG mice. What we can see here is that when we culture different ratios of T cells and target cells, that there is cytotoxicity of the allogeneic target cells as indicated by the dark blue lines, and then low toxicity from the syngeneic target cells as indicated by the green lines. Now in CD4 T cells, cytotoxicity is reduced only by class 2 monoclonal antibodies as indicated by the black line. In contrast, we see that with the CD8 T cells, the cytotoxicity is reduced by class 1 monoclonal antibodies only, indicated here by the red line. Now, these results indicate that the human T cells are not only differentiated, but they're also functional in humanized NSG mice. I would next like to highlight an example of an in vivo response. If you give the mice dinitrofluorobenzene, this will stimulate a delayed type hypersensitivity response, or DTH. We treated the mice with 2 doses of dinitrofluorobenzene to sensitize them, gave the mice a few days to rest, and then did a challenge dose of the dinitrofluorobenzene to the ear pinna. And you can see here there is a pro-inflammatory response as indicated by ear thickness. This response could be suppressed with a treatment of hydrocortisone as a proof of concept. These findings show that the T cells are functional in humanized mice and have a delayed type hypersensitivity response. Let's move on to another component of the adaptive immune system, which is the B cells. Mice do have some B cell functions, but they have limited class switching. For example, if you take humanized mice and immunize them with ovalbumin, you see that they do have an IgM response as indicated here in the white bars. However, they do not have a significant IgG response as indicated in the gray bars, and this indicates limited class switching. This is because they do not develop germinal centers, and unfortunately the reason for that is not completely understood. This may be due to human-specific growth factors or populations of cells that may be missing, such as follicular cells. This is an area of active ongoing research. Next, I would like to give just a few examples of researchers who have used humanized mice for various research applications. Please note that this is just a small sampling of research that we find particularly compelling, but we do curate a comprehensive list of publications on NSG mice on our website if you'd like to see how other researchers are using them. One of these applications is for regenerative medicine. This publication uses humanized NSG mice to test successful generation of blood progenitor cells from fibroblasts. The goal is to take a patient who has leukemia, extract skin fibroblasts, and transform them into human hematopoietic cells, and then engraft them back into the patient. This will hopefully allow them to repopulate all of their immune cells. Now, with different fibroblasts that were transduced with OCT4 to reprogram the fibroblasts, they become CD45 positive. Then you engraft this into the mouse and, using cell markers, explore what kind of cells are generated in the mouse. We can see that depending on the different cultures and factors that you can get different immune cell populations. For example, with FLT3 and stem cell factor, they got monocytic and granulocytic progenitors, and when cultured with erythropoietin, they get erythroid and megakaryocyte progenitors. So this is a useful platform to use when testing new culture and factor methods to promote regenerative medicine. Next, we're going to look at a couple of examples of infectious diseases. In these examples, the humanized mice are an effective model because for many infectious diseases, only humans can be infected. So small animal models are hard to come by. In this case, the researchers infect humanized mice with dengue virus, which is a mosquito-borne viral disease that has been rapidly spreading in recent years. They found that you can get a mosquito bite to deliver the virus into a humanized mouse, and this better models the actual disease than by performing an intradermal injection. We see that when delivered by a mosquito, the virus persists for longer in the blood compared to the virus injection. We can also see that when delivered by a mosquito, there is increased erythema severity compared to the injection. Furthermore, innate and adaptive immune responses were greater when the virus was administered via mosquito bite instead of the intradermal injection. So delivering the dengue virus via mosquito bite further improved the model, which is a big step to move closer into the context of human disease. The next research application is for the Epstein-Barr virus, or EBV. And this is one of the most common human viruses in the world that is most well known to cause mono. The goal of this study was to develop a model for EBV that would be relevant to humans. So researchers injected CD34 humanized mice with EBV intraperitoneally and checked for the presence of EBV in various organs. They were able to detect EBV in organs by about 4 weeks post-infection. And if they co-stained for EBV and CD3 T cells, you could see that they co-localize. Now, what was really interesting about this is that they found that CD34 NSG have effector memory T cells. They found human T cells would expand in the spleens of these mice, and they have an active memory phenotype. This suggests that this could be a strong mouse model for testing vaccinations against EBV. In this model, they also did a T cell depletion experiment. In this first panel, we see a control mouse spleen, a spleen that has been exposed to EBV, and a spleen that was depleted of T cells and exposed to EBV. Now, if you compare these tissues, you can see that mice that were depleted of the T cells have tumors in the spleen, the kidney, lymph nodes, and the liver. Now, this demonstrates that the T cells are what is keeping these tumors at bay. So therefore, when you put it all together, this mouse model recapitulates features of the Epstein-Barr virus infection and generates T-cell-mediated immune control that resists oncogenic transformation. Now, this may be a useful model to test the efficacy of EBV vaccines and to characterize the innate and adaptive immune responses during primary EBV infection. You can also use CD34-NSG to study adaptive T-cell function. In this experiment, the researchers did engraftment of B6 rag null skin onto NSG and humanized NSG mice. You can see that with the NSG mouse as a model, up to 20 days post-engraftment, the skin is well tolerated, even though the major histocompatibility matrix is mismatched. These mice are essentially so immunodeficient, they don't mount a response. However, you can see that in humanized mice, they reject this tissue transplant over time, indicating adaptive T-cell function. Now, as a negative control, if you do a syngeneic engraftment of humanized skin, this is well accepted by the humanized recipient. You can also see this reflected graphically here in both the B6 skin on NSG and humanized NSG skin on a humanized NSG mouse. These have successful engraftment, whereas the B6 skin on the humanized NSG have rejection. Using that same model, you can modulate how long the grafts survive by using anti-CD3 depletion of T cells to block the rejection. In this first figure, we can see that at both the high and medium doses of anti-CD3, we see that the skin is not rejected. However, with the lower dose, rejection is only partially prevented. These researchers also used anti-CTLA-4, which accelerated the rejection of the graft. So this is a proof of concept study that shows that the CD34 humanized mice engrafted with MHC mismatched skin can be used to investigate the ability of human therapeutics to enhance or suppress functional immune responses. In this final section, I'd like to touch on the immune oncology applications of NSG mice. We engraft human CD34 cells into irradiated NSG or SGM3 mice, creating humanized mice. Then we also put in a patient-derived xenograft, or PDX, which is usually a tumor fragment. At JAX, the result of this is what we call an oncohu mouse. This unique mouse model allows for the analysis of immunomodulatory therapeutics in the context of a human immune system.
One thing that we would want to first test in the OncoHu platform is the growth kinetics of the PDX fragments in the context of the CD34 humanized mice. To that regard, JAX researchers engrafted 3 PDX models in CD34 humanized NSG mice 12 weeks after they were engrafted with CD34 cells. In these 3 figures, you can see the growth of the tumor in total tumor volume in days post-engraftment. You can tell that for all 3 of these models, including lung, soft tissue carcinoma, and breast, that the growth rate of the tumors was similar between humanized NSG mice, as seen in pink, and non-humanized NSG mice, as seen in green. In this particular study, the HLA of the CD34 cells and the PDX were partially mismatched, suggesting that you may not need a complete HLA match for the tumors to grow. This is further supported by the fact that our own in vivo pharmacology group does not routinely HLA match PDX models and CD34 cells. Therefore, we expect that HLA matching is likely not needed for the PDX to grow. PDX growth kinetics can be variable depending on several different factors. Therefore, growth kinetics should be tested for each model before enrolling in a study. Most PDX models that we have tested grow well in the context of the human immune system, but not all of them. In the previous section on humanized mice, we discussed that there would be donor-to-donor variability in the CD34 cells. That is still true in the OncoHu platform. Therefore, several CD34 donors per experiment should be considered.
We next looked at tumor-infiltrating lymphocytes by looking at CD45 cells within the tumors, including those from breast, lung, and sarcoma. Now you can see varying levels of these CD3 and CD4 T cells, CD3 and CD8 T cells, and CD19 B cells within each of the tumors. So this indicates that there will be varying levels of tumor infiltration, likely depending on the characteristics of the tumor itself. So now let's look at some research examples using the OncoHu mice. In this example, researchers took a CD34 humanized mouse and engrafted it with a rhabdomyosarcoma cell line to test their novel therapeutic. In this case, they took readouts in terms of survival, which is this figure on the left, as well as absolute tumor volume, which is this figure on the right, and they did these analyses over time. You can see that they can increase survival or decrease tumor size depending on the treatment that they used. Let's take a closer look at what they found when they looked at the infiltration of different immune cell types, both innate and adaptive, into the tumors themselves after treatment. This was intended to demonstrate that the treatments are having a functional immune effect on the tumor itself. What we can see with this first treatment, which is this FcIL7, the immune cells were primarily natural killer cells, which are CD56, and macrophages, CD68, as indicated by staining. In contrast, sarcomas of the mice treated with either of these NHS IL-12 regimens showed a dense mononuclear infiltrate with natural killer cells, macrophages, and large numbers of both CD4 positive and CD8 positive T cells. This is once again seen across the board with staining and is also represented graphically on the right-hand side here. So these results indicated that different treatments will stimulate different immune cell responses within the tumor itself. So now that we have these OncoHu models, we can use them as a platform to test different immunomodulatory therapeutics for a human immune system. This would not have been possible with a standard NSG mouse engrafted with tumors. For this example, we took a triple-negative breast cancer tumor and engrafted it into CD34 humanized mice. This particular tumor has quite a high level of PD-L1, which makes it a good candidate for treatment with Keytruda, which is an anti-PD-1 drug and a checkpoint inhibitor. We see that the untreated tumor, which is this black line here, has robust growth over the span of 20 days after engraftment. For triple-negative breast cancer, the standard of care is cisplatin, making this a good positive control shown here in green. We do see suppression of tumor growth, with the tumor actually slightly decreasing in size over the 20 days. Now with Keytruda in pink, we also see suppression of growth. This suggests that the T cell population in the CD34 cells of the engrafted mouse is preventing tumor growth. To summarize, NSG and NSG-SGM3 are a proven platform for engraftment of the human immune system. We have seen that PDX growth is not grossly affected by HLA-type matching. We see that tumor growth kinetics are similar in humanized and non-humanized hosts, and that overall about 15% of PDX tumors fail to grow in humanized mice. However, this is not dependent on HLA matching. Human immune effector cells infiltrate human tumors in both humanized CD4 NSG and humanized CD34 SGM3. This is evident in CD4 and CD8 T cells, CD19 B cells, and CD33 myeloid cells. And finally, CD34 humanized NSG and NSG SGM3 that are co-engrafted with PDX tumors have been evident to respond to multiple anti-tumor agents. In this final slide, I'd like to highlight where on our website you can find some additional resources, both on NSG mice as well as humanized mice. Now, if you go to the strain data sheet of the NSG mice here, you will see a link under the detailed description highlighting resources for the NSG mouse model. From there, it will take you to some various resources for utilizing the NSG mice. For example, one thing I referenced earlier was a list of references that use NSG mice, and you can find that here in the Breakthrough Research Using NSG Mice. You can also browse through NSG variants by research area, view some of the flow cytometry characterization data, some of which I presented today. And finally, if you're intending on bringing these mice into your facility, we have some suggestions for housing and breeding. NSG mice are very immune deficient, so they do require some special considerations for care. This webinar was made possible with your support. JAX Mice and Services is dedicated to providing quality resources, flexible solutions, innovative technologies, and unsurpassed expertise and support to accelerate all aspects of your research. Every dollar you spend on JAX Mice and Services supports our shared research goals and sustains valuable mouse models and resources available only from the Jackson Laboratory.
Thank you for watching this video. I hope that this presentation has provided valuable information to help you achieve your research goals. If you have any questions about the information shared today, please contact us using the information on this slide. We would be delighted to support you with technical assistance.